Our next speaker is going to be William Foster, who's going to be talking about the cranial anatomy of Qianzhousaurus sinensis. So I'll just bring up your talk.
Thank you.
And there we go. Take it away.
Thank you very much. Morning, everyone. My name's Will, and I'm going to be talking about a part of my thesis from my master's by research degree that I performed at the University of Edinburgh. I was supervised by Dr. Stephen Brusatte of the University of Edinburgh and co-supervised by Dr. Junchang of the Chinese Academy of Geological Sciences. And so the title of this work is The Cranial Anatomy of Qianzhousaurus sinensis. So Qianzhousaurus is an aleoramin tyrannosaurid, which is kind of this weird morphological outlier subclade of the very derived end of the tyrannosaurids, which is tyrannosauroids, the most derived clade being the tyrannosaurids. And if you look there in the top right, there's a simple cladogram there, and you can see this subclade, this aleoraminized subclade, just coming off the very base of this group. And so the tyrannosaurids of this group full of these mega predatory bipedal carnivorous dinosaurs that terrorized the ecosystems of the latest Cretaceous. And we separate the aleoramins from the main body of the Tyrannosauridae for a few morphological reasons. Firstly, there's this very eccentric condition in the nasal rugosities. If you see in the top left there, that's one of the nasals of Aleoramus, which is one of the first to be discovered in lateral view. And you can just see how eccentric those peaks and troughs are. And that probably was would have provided a basement for some sort of keratin sheath. And there's a much more eccentric than what we see in normal tyrannosaurids. The second and probably most important thing we see in Aleoramins is this very long and low or longirostrine condition in the skull. You can see the maxilla there just below the nasals. That's also of Aleoramus. And in Aleoramins, the snout length tends to be twice as long as anything in the posterior skull. So these were dinosaurs with very long skulls compared to a lot of other tyrannosaurids. Thirdly, third important characteristic, but not really for this paper, we're focusing on the cranium, is generally quite a lightweight bore plan. The legs tend to be much longer than most tyrannosaurids of their size, and generally they're less stocky than things like Tyrannosaurus and Tarbosaurus. And just in the bottom left, there's a schematic map just showing where we found the current three taxons of Aleoramins. We've got Aleoramus remotus from Mongolia, Aleoramus altai from eastern China, and now Qianzhousaurus from southeast China. So from the very onset, Aleoramin research has kind of been plagued with this issue in that a lot of the characteristics that were trying to be cemented as their own subclade actually share them with juvenile tyrannosaurs in general. So quite rightly, early Aleoramin researchers were struggling to get people to accept them as their own subclade. If you don't have to worry too much about the labels there, just with peripheral vision, you can see that they tend to be quite similar to just juvenile tyrannosaurs. That's a juvenile tyrannosaurus on the bottom there, and then an Aleoramin skull on the top right. And you can see they're both very pneumatic. They tend to have quite weak cranial sutures anyway, delicate cranial bones, and the dentition is very similar. And you can just see in the top left there, that's both ends of the ontogeny scale with tyrannosaurids. You've got a juvenile there, and you can just see how the skull deepens and widens with age. And so the argument was that there's not really anything in these Aleoramins to justify them being their own clade. And that's where Qianzhousaurus came along. In 2014, it was immediately apparent that the material was of a later ontogeny animal. It was twice the size than Aleoramus. And so this little logarithmic graph was performed. And femur length in general is usually a good body size indicator for tyrannosaurids. And when you plot it against the skull length of Qianzhousaurus, you can see there on the graph, there are two tyrannosaurid specimens beneath it with exactly the same femur length, but that skull length is considerably longer than anything with that similar femur length. So they really are doing their own thing. And that was important. This paper established the animal in the phylogeny, but it didn't really go into much depth as to the osteology of the animal. And that's really what my thesis was. It was a big comparative description of the animal. And so we did pick up on a few more characteristics that were missed in the early paper. Generally, Qianzhousaurus has quite swollen corneal processes, and these would have formed the foundation of like hornlets and things like that probably expanded when the animal reached sexual maturity. We also see an intermediate dental condition. So you can see figure A there, that's Aleoramus altai, which is certainly a juvenile Aleoramus. And those tooth alveoli are almost rectangular, and that would have formed this quite mediolaterally thin zyphodont tooth, so a really recurved tooth. Probably it's nowhere near the banana-like, almost columnular teeth that we see in large adult tyrannosaurids. But figure B there is Qianzhousaurus, and you can see they're starting to take on that oval tooth alveoli shape. We also see evidence of higher ontogeny in the maxillae. Anatomy is quite hard without a pointer or anything, but I've done my best here to point out some of these features. So the maxillary fenestra in tyrannosaurids, as their skulls deepen and widen with age, it tends to move forwards in the skull, and it sits within this maxillary fossae. And you can see there's kind of a grading here. A is Alyramus oltai, B is Qianzhousaurus sinensis, and C is an adult Tarbosaurus. And you can see with later ontogeny, that fenestra starts to sit pretty confluently in with the outline of this depression. And you can see in Qianzhousaurus, again, it's quite intermediary. It's not really in the middle like it is in Alyramus oltai, but it's not sat right on the posterior region of this depression like it is in Tarbosaurus. So again, another intermediate condition there. So limiting factors. This dentition issue becomes a bit more apparent when you look at figure C there, which is an adult tyrannosaurid, and you can see that these teeth almost become columns. And although Qianzhousaurus isn't quite there, it is certainly more so than in juvenile tyrannosaurids. So although it's getting there, it's nowhere near this columnar condition that we see in large adult tyrannosaurids. We also have some open, well, not open, but still visible sutures that were probably closing in this animal as it aged, just like human children do. Their skulls will fuse. And you can see at anterior and posterior ends of the nasal there, you can still see that interneuronal suture. And although it is almost completely fused, it's still visible. And usually it's completely overridden in adult specimens. Some more limiting factors. There's a general fragility in the posterior cranium. Things like the quadrupedal and the squamosal, mediolaterally quite thin, and the sutures between them aren't very strong. So it sort of suggests this not structurally a very robust structure. We're also missing this postorbital flange, which in adult tyrannosaurids, figure C there is Tyrannosaurus, it comes out from the postorbital and basically cups the eye, and that probably would have increased skull strength. We don't see that in Qianzhousaurus currently. We think we see an incipient condition. So again, like the dentition, it's getting there, but it's nowhere near what we see in large adult tyrannosaurids. So what did we find in regards to jaw musculature and sort of anything that we can interpret for any sort of paleoecology? So on this figure, you've got A, which is again adult Tarbosaurus, B, Qianzhousaurus, and C, which is Alyramus oltai. And you can just see that changing condition between this adult Tarbosaurus and the syrangular shelf of Alyramins. It's almost this sausage-like structure in adult tyrannosaurids, and it turns into this more peg-like shelf in Alyramins. And this structure was very much involved in providing the basement and support for these enormous jaw-closing muscles that give Tyrannosaurus their characteristic bite strength in the literature. And what we're seeing in Alyramins is very different to that in large adult Tyrannosaurus. So there's definitely a cap there on the size of the muscles we could be generating, and that raises questions about the paleoecology of these animals. So what sort of hypotheticals can we pull from this anatomical assessment? So there's a phrase that goes around a lot in tyrannosaurid research, which is this puncture pull capacity, which would have allowed tyrannosaurids to experiment with extreme osteophagy. We know that they crushed and digested and excreted a lot of bones, a lot of horns. So these animals had access to marrow and things like that, which would have given them a major advantage against a lot of other theropods without that jaw strength. So are we seeing the anatomical foundations for that sort of behavior in Alyramins? We don't think so. And that's a combination of that general skull fragility and the lack of those big muscle attachment platforms that we see in tyrannosaurids. And then, so extrapolating from that, if they're not going to be using things like puncture pull feeding and stuff like that, what were Alyramins doing in the late Cretaceous that allowed them to exist alongside large body tyrannosaurids, which we know that they did. Alyramus was found in the exact same horizon as Tarbosaurus, which is a big, basically Tyrannosaurus-sized animal that basically stalked Asia at the same time Tyrannosaurus was was in North America. So tyrannosaurids are cool in that basically where they exist, there's very little competition in regards to other large theropods. We have things like dromaeosaurs and stuff like that, but they seem to dominate pretty much every ecosystem that we find them in. And that's probably because their anatomy changes so significantly with ontogeny. They tend to start off quite lightweight, and then they become stockier and more powerful generally. So you could have different predatory niches in the same ecosystem, just being occupied by different stages of ontogeny in tyrannosaurids. So Alioramens were not only competing in sympatricity with large-bodied tyrannosaurids, they were also competing with tyrannosaurids of younger ontogeny. And so they must have been doing something quite different. And we argue that a combination of a lightweight skull and a lightweight body plan in general and long cursorial hind limbs would have given Alioramens their niche for existing with large-bodied tyrannosaurids. I like this piece of art here, and I think it's probably quite reflective of what was going on in the late Cretaceous of Asia, where you've got these Alioramens harassing a large tyrannosaurid there. And I think that's probably quite right. And this is supported by the fact that there were a lot of oviraptorids and things like that, probably quite cursorial dinosaurs existing at the time, that large-bodied tyrannosaurids probably wouldn't be hunting. They were much too fast. And I think that's probably a niche where Alioramens can come in and fill. So what's next for Alioramen research? We really need to start approaching this with some quantitative methods. A CT scan of the skull would be great, and then we can perform real finite element analyses to really quantify a lot of these hypotheses that we have as to jaw strength and put that against large-body tyrannosaurids, just to measure accurately what sort of structural capacity these skulls would have. Another great idea would be a biomechanical analysis of that gracile bore plan, pit that against the more stocky condition that we see in large adult tyrannosaurids. And most importantly, as is the case with a lot of paleontology studies, we need more specimens, really. Qianzhousaurus was so important because it was more advanced than either Alioramus taxons, which were the first two to be found of the Alioramens. But it's still not late ontogeny. And what we really need is to complete that growth curve and find a Qianzhousaurus or an Alioramen, which is basically at a completely mature ontogeny, just to make sure that our extrapolations as to what skull elements were doing what actually line up with what it was like in life. So thank you to everyone that's given talks and everyone that will give talks. It's been really great this year, and I'd happily take any questions.
Brilliant. Thanks, Will. That was great. So we have one question in the chat from Jordan Beswick, and it says, is there any evidence of gregarious behavior in Alioramens?
There's no direct evidence, but I think we can, when we start to talk about what I said with this eccentricity in a lot of the basements for keratinous structures, these are obviously animals that were using their bodies for show, probably more dynamically than we see in large body tyrannosaurids. Those peaks and troughs in that basement were much more eccentric than what we see in other tyrannosaurids. So whatever keratin sheath, obviously not preserved, was growing on their skulls is probably more eccentric than what we see in most other tyrannosaurids. So they were, and that's an indication that they were at least more social, probably, if they're trying to communicate reproductive capacity and things like that with their skull morphology. So that's an indicator. Things like social structure and behavior is probably better interpreted from things like trace fossils and footprints, like it is for a lot of other dinosaurs, but it's really sparse for tyrannosaurids in general to find any actual traces of behavior like footprints. Yeah.
Okay, great, thanks. I think we're going to move on to the next talk now.
Excellent.
So I shall just stop this presentation. So our next talk is by Aodhán Gogáin, and it's microcomputed tomography of the aïstopods from the Jarrow Assemblage, Ireland. So I'll just bring up the presentation. Okay, take it away, Aodhán.
Thanks very much. Good morning, everyone. So today we're going to kind of move on to some slightly older tetrapods than the dinosaurs we were just looking at. So the Jarrow Assemblage preserves a diverse array of early tetrapods and fish and is located in the Leinster coalfield in southeastern Ireland. It's found towards the top of the Jarrow coal seam in an area where it thickens locally into a kind of channel. And it's thought that it may have been deposited in an abandoned oxbow lake, preserving an estuarine community of tetrapods and fish. The Jarrow assemblage is the oldest of the 4 major Pennsylvanian coal swamp assemblages that produce tetrapods, the others being Newsham in England, Linton, Ohio, and Niershany in the Czech Republic. Tetrapod specimens from the Jarrow assemblage preserve excellently articulated bodies, including scalation, and in some specimens there might even be the outline of the body. However, the specimens are highly compressed, and it's very difficult to make out a lot of detail in them, to the extent that even the boundary between the bone and the matrix can be difficult to distinguish. The tetrapod fauna from the Jarrow assemblage is dominated by mostly small tetrapods, including nectrodines and aïstopods. So aïstopods are a clade of tetrapod that sit on the tetrapod stem. They have these fenestrated skulls, elongated eel or snake-like bodies, and they lack any limbs and have these sickle-shaped pectoral girdles. Although the Jarrow assemblage doesn't mark the oldest appearance in the geological record of aïstopods, it does mark the first kind of described aïstopods. So the first aïstopods to be described were from the Jarrow assemblage and were described by Huxley in the 1860s. He named 2 taxa, Ophrodiprotodon brownriggi, which is represented by the 3 sketches on the top here, and Dolichosoma emersoni, which is on the bottom here. Now, Dolichosoma was actually transferred to the genus Dolichosomatites in the 1960s, as Dolichosoma was already occupied by a little beetle. And since their original description, there hasn't been a huge amount of research done on the Jarrow aïstopods, and that's again because the specimens are highly compressed and there's not a great amount of detail that can be made out of the fossils. And the reason why detail is hard to make out in the fossils is because bone has been totally altered. So this is a CL image with an aïstopod vertebrae on the left, and you can see that the original bone structure is totally gone and has been replaced by these coal pockets, which are surrounded by tabular apatite and sphalerite. To the right of this yellow line here is the matrix which surrounds these tetrapods, and this is also composed of coal, sphalerite with little crystals of apatite in it, and in some of the specimens you actually get apatite veins that kind of transect through the specimens. So you have these really compressed tetrapods that have a similar composition between the bone and the matrix, so they're not really ideal for computed tomography. We thought we'd give it a go anyway, and we got some real nightmarish CT scans like we have here. So this is the skull of an aïstopod, and it's very difficult to make out a huge amount of detail in it. However, towards the posterior, towards the top of it in this case, you can start making out some of the bone. And when you render these 3D models, they can actually be very informative, particularly when it comes to the arrangement of the dermal skull bones in the skull. So these are 2 models of Ophrodiprotodon brownriggi. So there's a larger, more mature individual in the top 2 images here, and a smaller, less mature individual in the bottom 2 images here. So Ophrodiprotodon have these anterior placed orbits. They have a closed temporal fenestra, which is closed ventrally by a contact between the quadrata jugal and the maxilla and jugal. And towards the center of the parietal, there is this kind of ossified bump or bulge, and this is seen in both specimens. This seems to be a unique characteristic to Ophrodiprotodon brownriggi, if not Ophrodiprotodon in general. In this specimen here, we have part of the cheek region along with the jaw, and this model was actually rendered from the CT slices that included that CT slice I showed earlier. So even in some of the most awful CT slices, we're getting some good 3D models out of them. What's really interesting about this specimen here is if you look at the jugal, which is enlarged towards the bottom left of the slide here, you get these little pits which are highlighted in dark gray. And these are interpreted as being part of the lateral line system. So they're lateral line pits or lateral line pores, similar to those seen in the jugal of Acanthus stiga. This specimen also gives a good idea about the variety of dentition in Ophrodiprotodon. So in the bottom 2 teeth in this slide here, you have these large marginal teeth which curve posteriorly towards their apex, whereas the third tooth beside them is a kind of smaller tooth which doesn't curve posteriorly and is part of these disarticulated row of teeth between the maxilla and the jaw there. These are interpreted as being teeth associated with the palate. However, which palate bone is unsure to which they attached. In this specimen, there's also some palatal bones, possibly a palatine, which has this kind of cross-secting row of denticles along it. In the lower jaw as well, we have these large or curved marginal teeth, but in this specimen here, around the large marginal teeth, you're also getting these much smaller, tightly packed teeth. Now, it's unsure whether these teeth are associated with the dentary, or could they be teeth from a coronoid or coronoids that have been displaced and are preserved just beside this bone? The presence of a coronoid or coronoids in the lower jaw of Ophrodiprodon is uncertain, and it's very difficult to actually make out a lot of the bones on the medial side of the jaws. The vertebrae in Ophrodiprodon are kind of typical of most aïstopods, so they are amphicolous, and they have this low, flat neural arch that's sutured, or not sutured, that's fused to the centrum. In Ophrodiprodon brown riggy, you see the transverse process doesn't contact the neural arch, and this is kind of characteristics of the vertebrae of Ophrodiprodon, because you see this in the Ophrodiprodon from the Jarrow assemblage. In the cervical area, so ribs from the cervical area are double-headed, while in the trunk region, at least by the seventh vertebra, you get these kind of K-shaped ribs. Now, these haven't been reported in Ophrodiprodon before, but you do get them in all other aïstopods, with the exception of the Mississippian Letiscus. Now, bones in the CT scans can be a bit of an eyesore. It can be kind of difficult to make out what bone is what, but the CT slices do give excellent images of the scalation in these animals. So here we have 3 images of Ophrodiprodon brown riggy, and on the right we have the pectoral girdles highlighted in red. And see along the trunk, along the bottom of the animal, there are these thickly packed gastrali, whereas in the cervical area, you have this kind of area, around the cervical area, you're getting this thick coating of these ovoid osteoderms. And these also run along the back of the animal, along the trunk, and also cover part of the skull, particularly around the temporal fenestra. TCD T90 is one of the original specimens figured by Huxley, but when you flip over the skull of the specimen and look at it, you can see that it's a bit different to that of Ophrodiprodon. It's much skinnier, and the jaws are much thinner as well. The skulls, or the bones towards the front of the skull, are kind of difficult to distinguish, but those posterior are kind of readily identifiable. And one major feature that sticks out is there's an open parietal finesse or foreman there as well. So this is a kind of key characteristic that suggests that T90 is not actually an Ophrodiprodon and is a new taxa. Dolichosomatites has a triangular or arrow-shaped skull with a closed temporal fenestra. It's long been considered to be related to kind of more derived aïstopods that have fused frontals and lack parietals and postparietals, but the presence of a paired frontal and parietals and postparietals in this specimen suggests that this affinity isn't actually really supported. And the scalation in Dolichosomatites is very similar to that in Ophrodiprodon, where you're getting these ventral gastrali along the trunk region, and in the cervical region, you're getting these ovoid osteoderms that go from the top of or the back of the animal down around the neck. So I added some additional postcranial and cranial characteristics to the data matrix of Anderson et al. 2003, along with some characteristics from Dolichosomatites as well. And this is the tree that was produced. So here you can see that Dolichosomatites is not really related to the more derived aïstopods that's been traditionally considered. It was traditionally considered to be related to Phlegidontia. And also that T90, which does appear to be a different species or different taxa from Ophrodiprodon, is actually closely related to Ophrodiprodon. Now, here I've kind of mapped out 2 different kind of groupings of aïstopods. So in the green-blue box, you have aïstopods with a closed temporal fenestra, and in red, you have aïstopods with an open temporal fenestra. Now, these 2 different groupings of aïstopods don't actually form any clades. However, there is an apparent faunal change at the boundary between the Bashkirian and the Moscovian, with aïstopod faunas being dominated by Ophidiopodon into the Bashkirian. And then from the Moscovian onwards, you get faunas that are dominated by Osteocephalus and Phlegidontia. There might be a possible link with this faunal change and climate change at the time. So going into the Moscovian, there is this sudden jump in global temperature. However, it'd be good to look at kind of other clades of tetrapods to see if this faunal change is also seen in them and if a link can be found between them. So working with the CT slices of the aïstopods from Jarrow it's been kind of, they're not the most nicest looking slides, but it's been very good in kind of actually describing the material which hasn't been really well described until now. And I just want to thank everyone for listening and your attention, and I'd be more than happy for any questions.
Brilliant. Thanks, Hayden. Great talk. So we've got one question in the chat already from Thomas Clements.
Yeah.
Tom says, great talk, Hayden. The taphonomy and mode of preservation is fascinating. Are there any soft tissues associated with these animals?
So people have suggested that there is soft tissue preservation. From the CT scans, you can kind of get the body outline from the scales, but it's very hard to actually, I can't see anywhere that actually looks like a body outline. Now, a huge problem with looking at the body outline in these animals when you're just looking at the hand specimen is that a lot of them have actually been kind of coated and are very glossy. So it's actually difficult to make out any real body outlines.
Okay, great. And then we've got a question from Sam Giles. Do you have any idea what the specimen with the open parietal foramen might be?
It's another aïstopod, but it's a new taxa of aïstopod because the postcranial material is pretty much the same as all other aïstopods, and it looks very similar to Ophidiopodon because it has this kind of closed temporal fenestra. The jaws are kind of hang loosely behind its head, and it has these anterior positioned orbitals as well, and it has the typical kind of aïstopod scalation tube.
Brilliant, thanks. There's a lot of nice comments in the chat, but I think we're going to move on to the next talk. So thank you very much.
Thanks very much.
So our next talk, let me just bring up the presentation. Our next talk is by Benjamin Griffin, Constraining Quadrupedal Launch, a range of motion in Coloborhynchus. Take it away, Ben.
All right. Well, hello everyone. So today I'm going to talk to you about range of motion analysis in pterosaurs. In this talk specifically, I'll be covering the Coloborhynchus and simulated soft tissue restraints that we've done for this fossil taxa. Now, I'm going to ask you not to share any of this on social media, as it's currently unpublished and is currently being worked towards that. So, well, it's down here. It's the next one. There we go. All right. So I'm sure everyone's very familiar with pterosaurs, but just in case, they were the first flying vertebrates that they evolved during the Triassic. Now, they are also the largest flying vertebrates, as has been very expertly shown in paleo art by Mark Whitten, which I'm sure many people have seen. Now, it is important to know for flight, launching is one of the most difficult parts. It's not only energetically expensive compared to regular flight, but it also has other issues where, as you get larger, your muscle impulse available for the launch does not increase at the same rate as your increase in size. This has led to some questions regarding how pterosaurs, which got so large, managed to launch at all. At this time, there are two hypotheses which get talked about, well, other than jumping off things, but they are the bipedal launch, which is a launch superficially similar to a bird, which is mainly just a hind limb driven leap followed by flapping, and the quadrupedal launch hypothesis, which was presented by Habib, which is more like that of a vampire bat, well, more or less, with a vaulting movement which has both the fore and hind limbs being used to generate the launch. So this quadrupedal launch was hypothesized as a method for how these pterosaurs managed to get to such large sizes. So this brings us to the aim of most of my study, which is to quantitatively evaluate these pterosaur launches. And in this case, we are looking at range of motion analysis in order to test if these were viable. So the first thing we needed for this study was a specimen. And in this case, it is SMNK 1133, which I am going to be referring to as Coloborhynchus, as that is what's on specimen drawer. Now, this may not be the most correct name for it, but in terms of what we're studying, that doesn't actually matter. What does matter is that this specimen is largely complete and undistorted, and it has a 5-meter wingspan, which makes it an excellent base for our model of a medium-sized ornithocurrid. Helpfully, it was also already CT scanned, which has made things a lot easier. Now, the girdles of pterosaurs are generally well conserved, which makes this a good case study generally for pterosaurs, as the range of motion likely won't be massively changed between taxa. Now, this specimen has been in the museum for decades and is well known in the literature, but because of its location and provenance, we are in discussion with some Brazilian colleagues about how to move forward with including it in the study. So anyway, now that we have our specimen, we had to make a model for it, which included adding a few additional pieces and duplicating a couple, just to make the full vertebral column as much as we could. And from there, we had to create the launch sequence for it. So we did this by approximating the launch poses in the actual specimen itself, and by examining the poses proposed throughout the literature. So this data was then all collated and applied to our Coloborhynchus model in order to make the sequence you can see on the bottom left, which goes from the quadrupedal base stance through either of the two launches and then into the flight pose. So these average poses can then be used for comparison for the range of motion analysis we'll be doing throughout this study. So, as I've said, we're doing range of motion analysis or ROM. The best method available for this is the ROM mapping methodology, which is essentially a method which takes a virtual joint in 3D space and rotates it through the full 360 degrees through all the different degrees of freedom, and will highlight any bone model that intersects with the other bones. So this is done using the workflow on the left, and the results for it they're much better than a normal range of motion study, as they allow you to check the interaction between the different degrees of freedom throughout the joint, instead of just looking at one degree of freedom at a time as you move through the animations. So, of course, we are working on fossils, and fossils require a few extra things when you're simulating them. And in this case, it is soft tissues. So soft tissues can both expand and constrain range of motion. And so we've had to look at both parts. So first off is the articular cartilage. Articular cartilage offsets the contact of the bony joint surfaces. So they will increase the range of motion. Now, for Coloborhynchus, we have no real good idea of how thick the articular cartilage was. And so to simulate it, we have used 3 thickness estimates from the literature and then applied the entire range of motion analysis through all 3 different offset simulations, as well as one without any offset as a osteological correlate. So the other one we had to look at was the connective tissues. These are soft tissues that will constrain your range of motion, and we needed to simulate those. And for this, we have used the ones that have the most control over the joints that we were studying. So when you combine these things, you end up with joint models that look like these. I am, again, going to remind you not to post the pictures of these as they are still being published. But as we stand, we have the pectoral girdle at the top, which is a hemicellular joint that is primarily constrained by the counteraction of different muscles, as opposed to a more common ligamentous capsule. So we know this because of the morphology of the scapulocoracoid is much similar to the more basal set, which is the muscular controlled. Also because there are only 2 confidently inferred ligaments, which is not enough to actually constrain a joint of this style. The pelvis, on the other hand, which is the one on the bottom, is a much simpler ball and socket joint, which is able to be constrained by the ligamentous capsule. So the main problem we've found with the ROM mapping methodology that we were using is that it only will determine your connective tissue lengths throughout your animation. So basically, as your animation goes through, these simulated ligaments are stretched, and it will give you these stretch values. What this means is we need a point to center on to increase into the range of motion, which is commonly referred to as the rest pose in literature on actual living animals. However, this is not a living animal, and so we can't find a rest pose. To get around this, we have utilized the triangulated minimum stretch pose. So this is the pose which has the lowest simultaneous stretch across each connective tissue that we had simulated. So, having combined all of these things together, we're able to create a lot of ROM maps. So I've only selected a few of them to show you today, but there's maybe 40 or 50 of them, so let's just stick with what we've got. So these ROM maps, they show the colored lines that are connected by those spots. Those are the different average launch sequences. And if they are within the gray shape, then they are considered possible or viable poses for the cartilaginous offset. Once you throw in the blue shape, which is the ones that are viable during the connective tissue offset, then you can check it combined. So for these, which are the pectoral girdle, you can tell from them that the Coloborhynchus range of motion requires at least some degree of appendicular cartilage in order to be considered viable. So for us, we went through a variety of different sizes, and the largest one, which I've shown you in the top middle, is the alligator, where you can see they're all within the range of motion, the range of ROM map, yes. Whereas for the connective tissue, which we simulated using the standard muscle simulation stretch values, which range from 20 through 40%, you can see that the launches become viable once you get into the 40% medial stretch, muscular stretch, sorry. So in terms of these pectoral girdle, they can be considered viable. Now, once we do the same thing for the pelvic ROM, things go a little bit different. So once again, we found appendicular cartilage was always required. So none of them were viable within the osteological ROM, which is the one without any offset. But even at our largest appendicular offset, which was the alligator again, the bipedal launch pose didn't fit inside the actual ROM envelope. So what this meant for the connective tissue ROM, which is heavily reliant upon the largest cartilaginous offset, as you can't get more than your cartilaginous offset anyway, it meant that no matter what stretch percentage we did for the ligaments we simulated, there was no way to make that viable. So in terms of the ligaments we've done, we've used them from the literature, and they ranged from as small as a 6% ligamentous stretch all the way up through a 56%. Now, on the other hand, our quadrupedal launch sequence, it again required a cartilaginous offset. However, once you got within 15% stretch of the ligaments, it was considered viable. So basically what this means overall is that the Coloborhynchus, at least using the average launches that we had, would be capable of a quadrupedal launch. However, it would not be capable of launching bipedally. So as I said, this is a model of a medium-sized ornithochirid that we were using. So we will hopefully be able to do this for a basal pterosaur and eventually the giant pterosaurs if we can get the material for it. But for now, I would like to thank my supervisors and collaborators. I do have to apologize. I did miss Peter Bishop off of this, and I'm very sorry for that. But beyond that, I'm happy to answer any questions.
Brilliant. Thanks, Benjamin. We've got a couple of questions in the chat from David Unwin. The first one is more of a comment. He says, many joints in pterosaurs have a tight fit, so the missing articular cartilage is perhaps less of a problem. And then his second point is, will this data also be used to analyze flight stroke and its limitations for both the fore and hind limbs?
So in terms of the appendicular cartilage, we tested 3 different ones. So that was the quail, which is a ground dwelling bird, ostriches and alligators. I only showed the alligator one in this for time and space constraints. But so there's actually quite a large difference in our articular cartilage work. But in terms of the flight, we could apply it to a flight stroke. It's definitely something we could consider doing. It's not something I have done yet, although technically I have the data for it. If you could find a set of flight strokes to compare it to, I could definitely do that very easily. But it's not something that's been done at this time.
Great, thanks. And then we've got a question from, where did it go? Oh, it seems to have just disappeared. That's okay. We've got a few more minutes, so if anybody has more questions. David Unwin has put a reply in the chat to you. We'll just hang around, see if there's any more questions. We've got a minute or so. Here we go. There's a question from Natalia. What fossil material are you planning on looking at for the basal pterosaur data?
So for the basal pterosaur, I have a surface scan of a Rhamphorhynchus, which hopefully I'll be able to convert into a 3D model by removing some of the taphonomic distortion, which will be hard, but basal pterosaurs are pretty hard to use anyway. There is also potentially a Durognathus we could use, but I have not yet managed to get that scanned.
Perfect. Okay, so thank you so much for the talk, Benjamin. We're going to now move into a break for the next 15 minutes. Just want to, before we end, just say thank you to all the speakers from this session. And a quick reminder that to get into the next session, you've got to use a different link. So you need to go back to the links that you've got, that you sent by email, and click on those to get into the next session. So thanks very much and enjoy the break.